Thank you very much. uh, so thanks everyone. I just want to take a few moments. I won't to kind of delay you from Laura's exciting work too long. I just want to give you a little bit of a brief intro to some of the exciting stuff that we've introduced recently at Brooker, particularly as it relates to the MALDI Imaging application and platform. And really, it's a culmination of a lot of our developments that we began back in 2016 with the RapidFlex, the first really commercial system devoted to MALDI imaging, and took some of that technology, also some of the fast proteomics work that we did with the timsTOF Pro, and kind of bundled all of that together and created the timsTOF Flex, which is really a fantastic QTOF TIMS trapped ion mobility system. And just shown here on the slide, the left side of the instrument is the proteomics metabolomics system, the timsTOF Pro entry point. The right side of the system is where we introduce the MALDI targets. And so it's a dual source system, and basically there's zero changeover. Just click the mouse button and you can switch between the two. And so all of these are kind of full capability. As I mentioned, you probably have heard timsTOF Pro already from a proteomics perspective. And then, now we've got that capability, the TIMS capability, and add that to with the MALDI source that we took out of the RapidFlex. So we've got the high pixel definition from the RapidFlex laser with the trapped ion mobility separation. And that kind of gives a little bit, this slide kind of shows it all. Basically, you have the MALDI separation here in this data set, M/Z in the horizontal, mobility in the vertical. And now we've got the capabilities of, separating things by collisional cross-section. And you can see here in the little inset here, within just a few hundreds of millidaltons, multiple images or multiple ions can be detected. And pulling out those respective ion images just shows you kind of the diversity of the distribution information that can be extracted. And so just this past year, we actually added to this system capability with a new technology called MALDI 2. Now, MALDI 2 means that, we've referred now to traditional MALDI as kind of MALDI 1, but MALDI 2 basically brings in a second laser into the system to do an additional ionization process to get better sensitivity. And you can really see some of the results here. If you look at this portion of the rat brain cerebellum, the left half was imaged with normal MALDI process, and the right half we activated the MALDI 2 laser. And so all on the same scale. And so you can see just this one particular ceramide that basically the MALDI 2 sensitivity is really enhanced compared to what it formerly was. So this kind of enhancement, it is compound dependent, and we see anything from, say, a 2x improvement up to 4,000 or 5,000x improvement. So it really kind of varies, and it's primarily noticed down in the metabolites and lipid regime. Not so much of an enhancement for peptides yet, but primarily noticeable for the smaller molecule perspective. But what this also gives us is we have the CCS aware. CCS, collisional cross-section, that's an inherent molecular property, just like molecular weight. And so once we measure this in MALDI, we can also measure it in ESI mode for LCMS. And now we've got two identifying bits of information for each peak. And now we have the ability to cross-correlate and bring those two datas together, and we've got that additional dimension of CCS to kind of enhance our confidence. And so we can do this in a couple of different ways. We can take our CCS and our M/Z of all of our panel of images and parse that through a metabolite or lipid database. And now we're not relying just on molecular weight as we've always commonly done in the past, but now we have the ability to integrate that with CCS for enhanced confidence. And so same like this, obviously the result is you've got, your CCS separated peaks, and now we have the ability to do those confident IDs. We also now have the ability that, since we have mobility separated as well, we have the ability to do fragmentation that is going to be very specific to one particular CCS value. So we get that built-in kind of enhancement. But the other aspect that this really brings into play is what we call spatial omics. And that is using the MALDI image to give us that unsupervised segmentation to tell us where a particular molecular region, a molecular phenotype of a tissue is that we want to maybe want to probe a bit more deeper with the LCMS approach. So this has been done in the past with histology, but as anybody that's followed imaging knows that, you know, molecularly we can define differences that histology often misses. And so here the concept of the spatial omics is to take your MALDI image, identify the molecular region of the sample that you really want to probe, and pass that over to laser capture microdissection for then a traditional kind of a bottom-up proteomics or a metabolomics kind of an analysis. So we get that, bring together that deep molecular information that you get from LCMS with that added spatial component from the imaging that gives you that very targeted region of sample to be probing. And that's kind of illustrated here, but more appropriately or more precisely illustrated in the data here from an application note. We have a breast tumor biopsy that a tumor region, actually you see here, segments into 3 subpopulations based on lipid profiles. And so each of those subpopulations can be sampled by laser capture microdissection, and then those removed material subjected to a bottom-up analysis. And then when you take all those protein IDs from each of those subtypes, plot out the protein activity from an ontology database, you can get profile activity profiles like the pie charts here that obviously are very different. So clearly the subtypes of a tumor, subtypes based on the lipid population, now we can get, you know, quite a bit more highly specific information like the proteomic profiles out of each of those to get that layer of information that you would otherwise miss. And so just as a quick summary here, basically the whole point of our spatial omics is why miss the spatial context of tissue microenvironment. With the bulk analysis, the traditional kind of omics analysis, grinding up the tissue and injecting it, basically you get signals from all cells, and then you got to sort out which cells come or which signals came from which cells. And then conversely, on a single cell analysis, your data is specific for the cell, but then you take it out of its native environment and you kind of miss a bit of its activity. And so by bringing together these components into the spatial omics analysis, you kind of can capitalize on both worlds of LCMS and MALDI imaging as well. And then bringing that together with our MALDI 2 to give the deepest molecular information from the MALDI perspective. So with that, I will thank you again for attending and for participating in today's webinar. And I will take the moment to hand it over to Laura.
I don't think I can share until you stop sharing, Shannon.
Oh, okay. Sorry.
It's okay.
I'm not used to Zoom.
Okay.
Cool. Okay, so while Laura is getting set up, it is my pleasure to introduce her for today's talk. So Dr. Laura Sanchez earned her Bachelor's of Arts degree in chemistry from Whitman College in Walla Walla, Washington, during which she completed an NSF summer undergraduate research fellowship in the Professor Phil Cruz's lab at the University of California, Santa Cruz. After completing her degree, she returned to UCSC to earn her PhD in chemistry in the laboratory of Professor Roger Linnington. And then she completed her postdoctoral research in the lab of Professor Peter Dorstein at UCSD in San Diego, which is how I know her. And there she established methods for probing and characterizing the metabolic exchange of between microbial communities, specifically those regarding cheese strains. She started her independent lab in the fall of 2015 at the University of Illinois at Chicago in the Department of Pharmaceutical Sciences. And just recently, she relocated and was promoted. So she's now an associate professor at the University of California, Santa Cruz in the Department of Chemistry and Biochemistry. Her team specializes in using and adapting imaging mass spectrometry and tandem mass spectrometry for small molecule analyses in complex systems. She was a K-12 Birch Scholar from 2016 to 2017, which supported the translation of the techniques she developed in Peter's lab to women's health. And she was a rising star in life sciences at UIC in 2019. I'm very happy you're here, and we're very much looking forward to your talk.
Cool. Thank you for that introduction, Vanessa. It's great to virtually see everyone. I wish I was there in person, but it is what it is. And I'm going to go ahead and apologize now. I have a puppy, and he seems pretty preoccupied with his chew toy, but just in case you hear squeaking, it's not me, it's the dog. Yeah, so with that, I'll just go ahead and get started. So as Vanessa said, the lab was recently moved back to UC Santa Cruz, where we are the mighty banana slugs. And today I'll be telling you about the work we're doing in ovarian cancer. And I think I always like to start this talk off, even though these things don't seem related, with thinking about what drives how we approach the system. So I grew up as a marine natural product chemist, and really, I think the marine natural products is an interesting system to be working in. And so what you're looking at here is the structure of didemnin B, which was the first marine natural product to ever be put into human clinical trials as an anti-cancer agent. It was isolated from a tunicate, shown here. And what they later learned, and didemnin is a growing story because people have learned things about this as time has gone on, is that it was a really potent anti-fetid. So, you know, these really structurally complex natural products have ecological roles. And I think what we later learned was that it wasn't actually ever produced by the tunicate. It was an alpha proteobacterium. And even going one step further, Jorn Peel's lab has shown that it's like spatially oriented within the tissue of the tunicate. So this is actually a fish image, not a MALDI image, of the didemnin producing microorganism in the host tissue. So I think, you know, what we've learned a lot about natural products is it really very matters, you know, the context in which these things are actually produced and how that might inform maybe how we could use them just beyond exploiting them therapeutically. So I think for us, this drives home a question of visualizing chemistry through space is important, and is there really a limit to the biological system we can apply this to? And so we really want to better understand the ecological context of small molecules to see if we can better exploit their therapeutic potential. And we're going to do that by keeping spatial referencing intact. And this is just a subset of some of the molecules we've worked on, but we're really like a chemistry-centric lab with hoping to connect it all. And so also, as Vanessa mentioned, we are both in Peter Dorstein's lab. So I think the other context for this talk is thinking about what tools do we have to look at chemistry through bacteria. So I removed the movie from this, but just to walk everybody through, because you'll see why this is important later when we adapt the technique, this is what everything kind of was built off. So in Peter's lab, we did a lot of MALDI imaging, mass spectrometry. And since this is a mass spec group, I'm not going to say the full name to save myself some time. But basically, we would grow bacteria or fungi on thin agar. So we would do about 10 milliliters in a standard size Petri dish, as opposed to 20. Coat it with a matrix using a stainless steel sieve, although now we know you can dry these things down, you can spray on them. There's a lot of different ways you can dry them, but basically you need to get the matrix on there. And then we simply pop it into the oven to desiccate it, which also adheres it to the plate. And then at that point, imaging kicks in where we can essentially irradiate at different raster points across the sample at defined spatial resolutions. And we'll generate a full mass spectrum at each point. So if you're doing untargeted metabolomics, this is great. We look mainly at the small molecule region, so somewhere between 100 Daltons all the way up to 2000. This generates a lot of data with an overall computational average of all the resulting data, which can be up to thousands of data points. That's where the magic comes in. So I think Vanessa maybe coined the term. This is our hypothesis generating tool. So basically in these untargeted microbial systems, this allows us to figure out, you know, was a signal colony associated, excreted, or is it just everywhere and part of the background as either the matrix or the actual media itself. And so, you know, my lab has gotten very good at also utilizing this same technology. And this is just an example of kind of like the bread and butter of what we do. So what you're looking at here is a Fusarium species next to a Ralstonia. So Ralstonia causes vascular wilt in tomato plants. And what you can see here is, even by eye, the fungi has a different response to this bacterium, whether it's the wild type or a knockout that makes a large lipopeptide. And imaging really allows us to pinpoint this directly, where we can see here, you know, the ionization of this molecule called bicaverin is very different. And what, you know, this also allows us to say is that bicaverin was described a long time ago as a weak antibiotic. And what this actually did was we could find this pigment in the survival structures of the fungi, and it was preventing the bacteria from having an endofungal lifestyle. So I think this is a really cool ecological role for something that turned out to be a horrible antibiotic, but it actually has a context in how these two soil microbes might actually interact with one another. And so I hope that everyone in this audience, mainly because Shannon already said his piece right before I started, is that, you know, imaging is a powerful tool in microbiology. But that was not the first use. I would say tissue imaging is far more popular. And I just like to point out this review where it's a nice kind of just subset of what people have been able to do with lipids. But most of the time this is done on tissues or tumors that are already there, versus the microbiology, we can grow these things and essentially run pseudo time courses. So this kind of allows us to see what happens before a final endpoint like a tumor. And what we really wanted to then do was kind of merge this technology. And the system we decided to do this with was high-grade serous ovarian cancer. So teamwork makes the dream work. Without Joanna Burdett and her postdoc at the time, Matt Dean, who's now an assistant professor at UIUC, and a former student in my lab, Catherine Zink, we basically got together and Joanna was like, you do all this really cool stuff with microbes. Can you do it in a mammalian system? And I was like, well, tissue imaging is a thing. But she was like, no, actually, high-grade serous ovarian cancer, the primary mutations actually occur in the fallopian tube and then move to the ovary, and the ovary is where you find tumors. So actually ovarian cancer almost is kind of the wrong name, because these things don't actually start in the ovary, that's where you just find the tumors. So really this is a spatially driven question, because this is also kind of the wrong way. Like normally things come out of the ovary into the fallopian tube, not leave the fallopian tube to head to the ovary. And so again, we wanted to know what really drives this, and can we intersect this interaction before physical tumors might occur on an actual ovary, because that's what you actually want to prevent, because ovarian cancer is the most lethal gynecological malignancy. It's typically caught very late stage, and because it's caught late stage, it has a poor prognosis. So if we can intersect it earlier, you have a much better 5-year survival rate at upwards of 90%. So we really need to look at these early events to see where we can actually interject and design interventions if possible. So what we started out, and again, I'm totally biased by the fact that I love small molecules because I'm a natural product chemist by training. What we really set out to do was, could we adapt what we knew about tissue imaging and imaging microbes to a mammalian system to look at the chemical exchange between the fallopian tube and the ovary. And our hypothesis was that this was going to be driven by small molecules, mainly because chemotaxis is a thing in the environment. If we go back and think about, you know, how microbes track through something like a tunicate, chemical gradients are everywhere in nature, so maybe they exist in the body as well. And how do we capture this? So this system is really, so now I'll walk you through what we actually do. So in mice, we're going to be using a murine model, mainly because it's very hard to get healthy ovaries from women. We only have 2. Usually you don't want to give them up early. So our system that we're going to be using is a murine model so that we have a constant supply of ovaries. And instead of calling it a fallopian tube, in a mouse, it's called an oviduct. And we're going to be using these healthy ovaries out of the mouse. And then we're going to be using an immortalized murine oviductal epithelial. So basically a murine fallopian tube cell line that has a P10 mutation. And since I was a chemist, I had to ask Joanna, P10 regulates the AKT pathway. And what they know about this cell line already is that if you remove P10, you get runaway cell proliferation and tumorigenesis. So we know that these cells, if you inject them into the mouse, will cause tumors. So it's a tumorigenic cell line, and we're using the healthy ovary. And so this is our setup. So now a movie may or may not play. Maybe I'll click start one more time. There it goes. And what you're actually looking at is how we've adapted what we knew about the microbial imaging to this system. So this is Catherine pouring in 1% agarose with about 50,000 cells into an 8-well chamber. So what you can see here is we've repurposed this 8-well chamber. It's now adhered to an ITO glass slide. And the little seed in the middle, seed-looking thing, is actually the ovary. So we place half of a murine ovary into the wells and then pipette the agarose cells and the media around it. So we're going to allow these things to have chemical exchange for 4 days while they incubate before we actually then image them. And that took a lot of optimization. I've totally just trivialized probably like a year of Catherine's life, but there it is. And so this is just what this ends up looking like. So these little 8-well chambers, as long as the trick to this is you just have to release them from the sides using a little spatula. The whole thing just pops right up and off. So you get these nice, even agarose pads. And then just like the microbial imaging, we subsequently pop it into the oven and dry it down. And you can see these dry down nice and flat and are well adhered to these ITO slides. We then coat them with a matrix using a TM sprayer. And then this is the actual fun bit, right? So what are the results? So I think the nice thing about the 8-well chamber is that this allows us to control for a bunch of different conditions. So we like to control for both, just media, and then both gene and cell specificity. So we have a Mohs scrambled line and then a murine ovarian surface epithelial line, just to make sure that it's not just a cell touching an ovary signal that we're going to prioritize. And again, the co-culture is really what we're interested in. What signals appear or go off when we have the healthy ovary next to these tumorigenic cells. And just to kind of convince you that we think that this works is, this is just a subset of some of the signals we saw. And you can see that there are differences depending on the condition it was grown in. So there are certain signals like M/Z 190 that appear to be totally gone anytime you have the MOP10 cells there. So we might compare it to where you see the ionization across the sample normally. So we might argue that this is a media component that's being totally consumed only in the presence of the MOP10 cells. We see other signals that appear to be associated with the ovary, and then other signals appear to be on/off depending on whether it was a co-culture or not. So we think, given these discrete spatial distributions, that we indeed have a really solid working system, and we're actually measuring the chemistry across the system. So the first thing we did was to use skills software to actually statistically prioritize which signals were upregulated in this MOP10 ovary co-culture across multiple replicates. So we usually like to do this 3 or 4 times before we move ahead. And what Catherine did then was to really kind of zero in on this M/Z 170, mainly because it was the most strongly statistically upregulated of all the signals we had. And when she did some dereplication using the human metabolome database, norepinephrine actually kind of popped out as an immediate hit to us. And so you might be asking yourself, why was this an immediate hit? Mainly, norepinephrine had a really well-established role in ovarian cancer. So it's known to increase VEGF, cell invasion, chemotherapy resistance. It is also known to be stored in the ovary, in the granulosa cells, and it peaks before ovulation. And I think really interestingly, so a lot of this research came out of MD Anderson from Anil Sood's group, where they were looking at stress-related catecholamines. So they were screening these things at 1 and 10 micromolar. They described all this with norepinephrine, and then went back and actually did a retrospective study in women and found that women taking beta blockers when they had ovarian cancer lived 60 months longer. So there was already an immediate translational value, but the question is, what is the source of norepinephrine in the system? And so to answer that question, we actually slightly redesigned our system, because if you recall the imaging I just showed you, it doesn't actually give you that information. So we had the ovary and co-culture with the cells, everything was dispersed, but it doesn't actually tell us what is what. So to redesign the system to ask a spatial question, so was it the ovary or the cells, we actually redesigned this using a divided chamber. So what we have now is, what Catherine takes is a sterile basin. She carefully measures these, sorry, the dog is going crazy now. Okay. We carefully measure these things and then wedge them in. So what we're going to do this time is to pour half of the amount of cells and agarose that we would have used for the full chamber into 1/2 of the well. Let that set. And so you can see here, she's just mixing all these things up. And then she'll pour that on 1/2 And then the nice thing about these little plastic removable dividers is that they can be removed. So what you're just seeing now is what it looks like to pour half of these, and you'll see in the takeaway, you get a nice little triangle. So after we have that, what Catherine then does is to place the ovary. So we're going to physically separate the cells from the ovary, and then surround that with media and agarose, but no cells this time. So the nice thing is the agarose pads will dry together. So even though, you know, they're poured separately, they're allowed to essentially solidify together, but everything is physically separated. And mammalian cells do not move through agarose. They do not have the correct cellular machinery to do so. So this time the setup to ask what the source of norepinephrine is, here we have the ovary, the media, and then the cells. And again, we're going to incubate for 4 days because that's what we did last time. And what we can see here then when we keep directionality in mind is where did the signals come from? So again, this M/Z 170, we think if this is the ovary and the cells are here, that this signal appears to be excreted towards the cells. So this tells us the ovary was the source of norepinephrine, and that directionally it appears to be headed towards the cells themselves. So the other thing we did then to help validate this was to actually test this in our own system. So Joanna's postdoc, Matt, did an invasion assay, and we looked also at mutant P53. So most cases of ovarian cancer have a P53 mutation as well as the P10. And what we can see here is that at 10 micromolar, norepinephrine increases statistically invasion of these cell lines. So our results totally track with everything that Anil Sood had previously published. And we think that this is really how these cells, they're essentially hijacking normal chemistry. And so we think really here, the ovary is supposed to have norepinephrine, but the fallopian tubes are doing something to cause it to release that. And then we already know about all those other properties. So now the question becomes really, is this the chicken or the egg? What did the fallopian tube cells release in the first place to cause the ovary to do that? Because if you'll recall, none of the other conditions really cause this upregulation or release of norepinephrine from the ovary. And so we call this signal factor X, and that's what we're working on now, is figuring out what's going on here. So we used factor X as just the term until we know what this is. We rationaled that it was a soluble factor, so we knew it had to be from the cells and that they would just be producing it because the signal came on in both the non-touching and touching or divided and non-divided chamber setup. And since I'm a natural product chemist, what we then like to do is called bioassay guided fractionation. And so to do this, we basically took our cell-free supernatant, spun it down to generate a protein fraction that we retained. And then we took essentially the flow through and partitioned that using chloroform and water. So we have essentially what we believe is a lipid, a polar compound, and then just the media itself to check what's going on. And so to do bioassay guided fractionation, you have to have a solid readout. And so for us, we rationaled that presence of norepinephrine was actually going to be a nice readout for us. So we took our same imaging mass spectrometry setup where we have the ovary, and this time we're not going to use the cells. We're going to take these cell-free extracts that we've generated to treat the ovary to see if we can get it to release norepinephrine. So again, this is just an optical image of the cartoon version of what I just showed you. And here you can see when we image this that it appears that the protein fraction with the ovary causes the ovary to release norepinephrine. So like, great, okay, it's a protein. And at this point I was like, oh man, like now I actually have to do proteomics, which is fine. But the next thing we did, since the word protein is a huge word, is to kind of try to further rarefy which type of protein it might be. So to do that, we use these spin filters again. So we have basically big proteins, medium and small, to see if we can parse out which kind of protein it might be. And again, we put it back into our bioassay guided fractionation system and found this time that it was really the 3 to 30 kilodaltons, maybe the 30 to 50, because these molecular weight cutoffs aren't hard fast. So maybe it's a protein right around 30 kilodaltons is causing the ovary to release norepinephrine. And just like true bioassay guided fractionation, we appear to be losing bioactivity as we continue down, which is normal. So the next thing we did was actually to now do an actual bottom-up proteomics experiment. So we called our good friend Stephanie Colonna and her student at the time, Melissa Pergandy, over in the chemistry department, and Catherine was able to go over there to learn how to do some bottom-up proteomics to see if we could identify this. So we're going to use our same cell lines, the MoP10 with the scrambled mutation compared to the Mo scrambled. And we took these fractions that had come out of the spin column. So they were already rarefied. We're not doing the whole lysate or anything like that, and just compared them from an identification standpoint. And what kind of came out, because we were looking at abundance differences, was a pretty small list. So there were 27 that were shared, 19 unique. And again, this table that you're looking at is really driven by abundance. So what you can see here is there's really only a handful of things that were more abundant in the MoP10 cell line over the Mo scrambled from this rarefied fraction. And so we could have tested all 4 of these things if we felt like it, but what we also knew and had at our advantage was that, again, remember I told you we're working in a murine cell line. We also knew that this worked in a human cell line called SCOV3, because that's actually all the work Anil Sood's lab had done. And we have the same invasion assay, so we knew that norepinephrine increases invasiveness in SCOV3. We also know that it really blows the signals out of the water in the imaging compared to any other human cell lines we've used. And it does the same 3 to 30 kilodalton fraction, bioassay guided kind of fractionation as the MoP10 cells. So we think what we wanted to do is actually, instead of just moving on with the Mo and the Mo scrambled, we actually wanted to throw in a cross-species validation using the SCOV3 cell lines. So that's what we did again. So Catherine did bottom-up proteomics, repeated it in the same way, and found that there were 14 proteins that overlapped between the 2 conditions in all of our experiments. And again, when we look at what these proteins were that were really unique to this, again, this protein called SPARC comes out. And so we really kind of settled in. If you recall, SPARC was also in the previous table. We really kind of zoned in on this. I don't think it's glycerol 3-phosphate dehydrogenase. I just think that's an artifact of doing bottom-up proteomics sometimes. And so we really wanted to move forward and validate whether it was SPARC. So we actually bought recombinant SPARC and just plopped it onto the ovary and treated it, and found that the recombinant SPARC does not cause the ovary to release norepinephrine, but that was the simplest thing we could do before COVID hit. And really, if you think about it, that would have been it would have been awesome if that worked, but in reality, there's a lot going on for why this might not have worked. There could be post-translational modifications or other interacting partners for why a recombinant version of SPARC wouldn't just work on top of the ovary. And so now you also might be asking yourself, well, Laura, what is SPARC known to do? So SPARC really upregulates the production of these collagenases. And if you're not familiar, the ovary is actually mostly collagen. So what we think might be happening is that the fallopian tube cells are secreting SPARC. SPARC is making it to the ovary. And then if it's remodeling some of the outer layers of the ovary, that's actually where norepinephrine is known to be stored in those granulosa cells. So we think it might be indirectly increasing the amount of norepinephrine, which is why we're now redesigning the experiment to do in an interactome, which might again also explain why the recombinant SPARC didn't just work when we treated it with the ovary. So I hope at this point I've convinced you that ovarian cancer is really a multicellular organelle problem, and that really designing a multipartate solution might be a better outcome. Because I think now, if you can block essentially both sides of this communication, or even think about how nutrients were removed from the system, I think that might be a better way to think about how we design earlier interventions or start to think about. And so I hope now that I've convinced you that, and I mean, everyone probably knew this except for me, but chemical communication extends far beyond small molecules. So I think this project really got us thinking about other types of biomolecules. So now I've got a little protein here in my original schematic. And so the question that we then asked ourselves is, have we been a bit too narrow, and can we actually exploit these other biomolecules in the same system? And so to do this, we actually thought about repurposing the pap smear. So can we read out biomolecules? And again, I'm going to really say we're riffing off Biotyper here, and the idea that small molecular weight proteins, and in Biotyper, it's really running off the 16S ribosomal proteins, because we know that they're highly abundant, so they're in high copy number and can be ionized in this range. Can we do the same thing, but for women's health? Because again, much like ovarian cancer, I think the idea here is that there's just no screening tools. So we always find it really late, and that's because there is no routine screening for it. But if you think about proximity and space as being important, then we might be able to repurpose something like a pap smear, which is in close proximity to where, or as close as can be, proximity to where tumors are actually going to be growing. And again, can we use these the cells or the cellular debris to detect ovarian cancer? And so the first thing we did was actually figure out how is this workflow going to work. So people have essentially biotyped mammalian cells before, but we really wanted to find a rigorous protocol. So that was done by Valia Pechikova in my lab. And what you can see here is we get a nice readout from mammalian cells. If you just compare two different mammalian cell lines, so OVCAR is a human cell line versus a murine cell line, that we do indeed see differences. Sorry, I thought he was chewing the couch. And when we mix these two cell lines together, you can actually see that you get distinct features even as low as 1% of a OVCAR in a murine oviductal epithelial background. So the MALDI fingerprinting is good enough to differentiate cells even at just 1% mixtures. And this was just a nice proof of principle that this could be done, and that mammalian whole cell fingerprinting gives us discrete features. So we moved forward with a mouse model study. So this was done by Melissa Gailey in my lab with the help of Ali Young in Joanna's lab. And what we did was we took 5 mice, injected them with OVCAR 8 RFP, monitored them for 8 weeks, and we took a vaginal lavage, which is like a murine pap smear. They don't make tiny brushes, so it's the closest thing we could get to a pap smear in a mouse, and pap smeared them every week. And so what we could tell then was by IVUS imaging is were tumors growing, and then how did that compare to the MALDI fingerprinting that we were going to take? And so here, this is just one mouse as an example, but tumors did indeed increase over time across all 5 mice. So the study was working. And when we humanely ended the study, they did have cancer on their reproductive system. And so again, to trivialize a large amount of work from the student here is we have 5 mice, 8 weeks, and we took 24 technical replicates for each lavage. So you might imagine then that's a lot of data points. And so we really thought carefully about how we were actually going to handle the data. So this is just to summarize that we think about it and we do it, and the code is available, but we think very carefully about how we handle all this data. And so the first thing we did was to ask ourselves how similar or dissimilar from a broad overview were these fingerprints that we took? Because again, this is going to be something like 1,000 different data points. So when we did a very simple cosine similarity matrix plot, what we could see here is that the weeks compared to one another, and we started with things that were similar across all 5 animals. So what we can see here is at the end of the study, if we compare week 7 to week 7, you get a very high cosine similarity score. And just to remind everybody, a cosine score of 1 is very similar, 0 is dissimilar. So the higher the cosine similarity score, the more related the spectra are to one another. So when you have a very high tumor burden, all of the biological animals look very similar. And I think the thing to point out is here, even at week 1, the animals still look more similar than dissimilar. So it's not like each animal is its own very individual spectral signature. There is commonality enough that this cosine score was still probably somewhere at 0.6. So it's still more similar than dissimilar. And just to show you, this is what the spectra actually looks like, because some people like to see that. And this is just an average of the technical replicates at each week for 1 mouse. So the next thing we did was to ask ourselves, okay, so we have good similarity. So what were really the signals across all 5 mice that were kind of driving this? So what occurred if we just took week 1 and week 7, what happens? So what we can see here is over time, 117 features were statistically upregulated across all 5 mice over time. And so if we just look at this last endpoint and pull the top 10 based on their p-values, we can see here it actually all kind of occurs in a similar range. Which is interesting. And we can also do the same thing. So we can ask which signals were actually downregulated. So we had 97. And again, if we just look at that last 10 endpoint, interestingly, we see that they're in a range that's about half the weight of everything that was actually upregulated. So we can see both things that are coming on and coming off. More come on when you have cancer. And then just to convince you that this is real, this is a composite of the spectra. So you can see signals do indeed occur. They show up over time and they decrease over time. So this is the real data. And now the next thing you might be asking yourself is, well, Laura, do you just have a glorified sick mouse test? So we asked our colleague, Natalia Nieto, if we could borrow some of her mice. So we actually did vaginal lavages on these mice that had non-alcoholic, they basically have like a fatty liver disease. So it's not ovarian cancer. It's obviously very different, where they have liver inflammation. And what you can see here is the signatures were different. So we don't think we have a glorified mouse disease, that each peritoneal disease might actually offer its own unique signature, probably. And so then the question became, this is always what reviewers want to know, is fingerprinting is great. I think we can definitely use it to tell these things apart and watch disease progression over time, but what are these signals? Because I believe Karsten Hopsgrove has postulated that these are histones in this region or different things. And so again, we called our good friend Stephanie Colonna, and this time her student Mi Nguyen. And Gordon this time learned how to do some bottom-up proteomics to see if we could actually identify these, because working with small proteins, I think most people in this audience are well aware that it's actually kind of difficult, because you can either just digest them too much or not enough. But we did see here from healthy tissues is that we can get signals and identify proteins in this small molecular weight region using a slightly modified setup. So again, it was 57 IDs were found from a pooled lavage sample, which is not a lot, but I also think if you consider the sample, we're dealing with proteins that are already soluble and there's no tissues involved. It's just cells or cell debris. So I think given the sample, this seemed really reasonable. So Gordon just did a PCA of tissues. You can tell them apart. So that's great. We think this system works and can be used to differentiate things in this region. And then this is really kind of hot off the bench work right before we left Chicago. He took our vaginal lavages, so those same lavages we've done all the MALDI work on, and then did an identification workflow. And so what we found is 14 signals popped up in the right protein range with good sequence coverage using bottom-up proteomics. And then what Gordon went through and actually did was, do we see anything in this same range in the actual MALDI data? And so excitingly, we see one that's actually a perfect hit. So cystatin A. So what you're looking at here now is actually all of the raw data out of the first half of the study at 11,006. So we can see here there's a signal, and this is the back half of the study, all the raw data, the signal increases over time. So it's really cool. We believe we've tentatively identified cystatin A out of our data as being something we could identify using bottom-up proteomics that was statistically upregulated over time in the MALDI data as well. And so now we ask ourselves, what the heck is cystatin A? So it turns out it's a cysteine protein inhibitor, which is interesting because if you recall, proteins that were essentially twice the amount of weight as the ones that were downregulated were the ones that were upregulated. And so being a protein inhibitor, maybe this is part of that. So we're essentially maybe capturing some of the physiological response. It has a known role in other kinds of cancers, although the role is very tentative, but we're pretty excited now to see if we can further figure out what its role might be in the context of ovarian cancer. And we've also sent this over to our collaborator, Judy Sue at the University of Arizona for her little sensors to see if she can have better modes of detection than we can, or to maybe multiplex it with the MALDI in the future. So I hope at this point now I've convinced you that these microproteins or small molecular weight proteins are an underexplored biomolecule class, but maybe they can be used to gauge host health. And so I think what my lab has now learned is, all sorts of biomolecules are really important, and it's really thinking about how do we critically measure these things to see if we can actually impact human health. So the next thing we're doing to translate this is actually working with human tampon samples. So we were able to get these from Anthony Godwin's lab, and Gordon has extracted these, and just at first glance, like again, hot off the bench, we think we see differences from real human samples. So their diagnoses are blinded to us, but they're they've been classified clinically before surgery. So we're really excited to see if we can actually translate what we've learned in the murine system to the human system. And again, I think what my lab is really thinking about now is how do we try to image and merge, you know, the small molecule with the biomolecule and the complexity of the different kinds of systems, because I think that's going to be really where we push forward with results. And so with that, you know, again, teamwork makes the dream work. So Gordon is currently working on the project. This is probably like the last time we were all allowed to stand next to one another for a team photo in Chicago. This is the new view in Santa Cruz, but Gordon is working on the project currently. Valia did the whole cell fingerprinting. Melissa did the mouse study, and Catherine pushed forward all the imaging that I told you about in the first half of this. We have funding from the NCI. As Vanessa mentioned, I was a Birch Scholar, so this really allowed us to translate what I knew in bacteria and fungi to mammalian systems, and then funding from the Research Corporation for Science Advancement as well. And with that, I'd be happy to take any questions.
Great, thanks, Laura. Does anybody have any questions?
I have a question.
Sure, go ahead.
So, hi, Laura. I'm Natalie. So, in your ovarian assay, did you have to use intact ovaries, or are they pieces of ovaries?
Yes, so we actually cut the ovaries in half for this, so we can get more bang for our buck. And we also think that that simulates ovulation somewhat. So, you know, it is normal for the ovary to rupture during ovulation. So we also think cutting in half partially mimics that.
I see. And so the norepinephrine, if you had an intact ovary, do you think you would have seen the same norepinephrine signal?
I think so. Yeah.
Or do you think the structure of the ovary would somehow?
Yeah, they can be cut in half. I think so. Joanna's lab they use half ovaries really frequently for a lot of their other assay type things. And we did try whole and half cells, or whole and half ovaries, when we were optimizing it. And we basically found no differences whether we're using the whole or the half. It's just the half gives us, you know, we can use less mice if we're using half.
And
yeah.
Another technical question was, why didn't you do the proteomics on using a quantitative assay?
Yeah, we could have. So we were repurposing samples that we'd already were using. So we'd already completed the mouse study basically a year before. And we really have to pull everything to get enough signal. So I think that's not to say we couldn't go back, but we also don't know, the samples weren't totally fresh. So I think it's something we're probably going to need to do it again anyway. We would do it a different way next time.
And maybe even fractionate the fractions with the groups with higher resolution to be able to correlate better.
Yeah, I think we would change it if we could go back, but we were trying to just repurpose a lot of the samples that we already had, because then it was also the same exact sample we had for the MALDI, but we only pull maybe, like on a good day, you get 20 microliters of a vaginal lavage back out of a mouse. So these are very sample limited.
Oh, I was thinking more in terms of your little well assay, your little square well assay.
Oh yeah, we were also trying to get someone to send us some of that new matrix that's like really good for catecholamines that they've been using in Per Andren's group, but they're patenting the matrix, so we haven't gotten any yet. Because yeah, I think now that we have a different instrument coming, so all that was done on an Autoflex LRF. The installation of the timsTOF Flex is happening this week, so I think we're also thinking about, you know, how do we use these different capabilities that we'll have access to now.
Any other questions?
I have a quick one related to the comment you just made there. You talked about the stuff from Per Andren's group. Is that the FMP10, the one that like functions as a matrix as well as
Yeah.
Yeah. All right. If you get that going, let me know, because I've been interested in trying that out. We have some studies with neurotransmitters that we've been interested in looking at. So
Yeah, I mean, I might just try to convince a chemist here to make it for me, because
I
I
thought
don't.
about that too. It's not that bad.
No, it's the purification might be a little tricky, but I'm hoping he can just send me some. Because if it doesn't work, then we don't have to go through. You know, if it works well, that then it's worth the synthesis. But
Definitely.
yeah.
Cool.
Any other questions? Well, if there's no more questions, thank you, Laura, for a wonderful talk. Really impressive work, and I'm excited to see where it goes.
Thanks.
Thank you.
Thank you, Laura. And hopefully we'll see anyone who is interested in their next talk on April 8th.